My name is Sandra Citi, and I'm the senior author of the paper entitled Cingulin and paracingulin tether myosins-2 to junctions to mechanoregulate the plasma membrane. And I'm speaking on behalf of the first author, Dr. Florian Rouault, a senior postdoc in my lab. The players here are cingulin and paracingulin, which are two proteins present in the cytoplasmic phase of apical junctions of epithelial cells, tight junctions and adherent junctions, and myosins. Myosins are essential motor proteins that bind to actin filaments to generate force. In epithelial cells, there are three types of non-muscle myosins, three isoforms, A, B, and C. But nothing is known essentially on the mechanisms that determine the localization of one or the other isoform of these myosins at junctions or in other parts of the cells. So in this paper, we examine how cingulin and paracingulin are involved in the localization of the myosin and whether they interact directly. And using purified proteins, we show that both cingulin and paracingulin interact directly with specific isoforms of non-muscle myosins, and this 2B for both cingulin and paracingulin, 2A for paracingulin, and myosin 2C doesn't seem to bind strongly to either of them. The binding is mediated by the C-terminal coiled-coil regions of cingulin and paracingulin that bind preferentially in an antiparallel fashion to coiled-coil regions of myosins. This interaction is important physiologically. So we used different experimental approach. In one of them, we knocked out cingulin or paracingulin or both in epithelial cells, and then we looked at the presence of the myosin at the junction. And in another approach, we overexpressed cingulin or paracingulin in double knockout cells to see what would happen to the myosin. In short, the results are that if you remove cingulin, you lose a lot of myosin 2B at junctions. If you remove paracingulin, you don't see many effects, but if you overexpress paracingulin, you have an accumulation of 2B and 2A, and if you overexpress cingulin, you accumulate more 2B. And if you do these experiments of exogenous expression using truncated mutants that lack the ability to bind to myosin, you don't rescue. So this indicates that the direct binding of the cingulin coil-coil, the paracingulin coil-coil, to the myosins recruits them to the junction. And this is the first time specific mechanism of recruitment of these isoforms to junction has been identified. Now, what's the consequence of this for the function of the cell? We have looked at different things, and I just can summarize a few. First of all, the amount of ZO1, a protein that binds to cingulin, is increased when cingulin recruits myosin. Second, the shape, the tortuosity of the tight junction membrane, which in mDCK cell is very tortuous, is completely abolished in cells knockout for cingulin, a bit less for paracingulin, and completely abolished in double knockout cells. And if you rescue, again, with cingulin or paracingulin, you recover tortuosity, but not if you rescue with the mutant that do not bind myosin. Second phenotype is the stiffness of the apical membrane. The apical membrane has a certain stiffness. If you remove cingulin, it becomes floppy, as if a trampoline has lost its attachment to the side. And again, this phenotype is dependent on myosin binding region. And finally, for paracingulin, we can see that losing paracingulin induces a fragmentation of the protein complexes at the adherens junction. And again, this is dependent on the transmission of force, which is mediated by the myosin 2B binding to paracingulin and connecting it to the belt. So in summary, our results show that by binding to specific myosin isoforms, cingulin and paracingulin couple the actomyosin cytoskeleton to the apical junctions to mechanoregulate the plasma membrane. And of course, this raises interesting questions for future studies also in animals, and using purified proteins to look at the effect of cingulin and paracingulin on assembly of myosin filaments and the regulation of this process.